Welcome back, friends. Welcome back in another video tutorial for microbiology with learning. So we are discussing about PCR techniques, right? In the very first video I have discussed about the basic principle of PCR. Then we have discussed about the reverse transcriptase PCR, and today's installment is about the part 1 of the real-time PCR, where I am going to discuss about the mechanism how the real-time PCR is going on, and in the next video I am going to discuss with you about the PCR cycle and CT values, okay, by which we can quantitate the amount of target DNA that is being amplified. Okay, so let's start. So comes what comes in your mind when you hear the term real time PCR? Okay, so it simply means that during that PCR technique, it allows the scientist to view the amount of DNA increasing. Okay, that means it provides the scientist the opportunity to absorb the increase in amount of target DNA during the PCR technique. Okay, that is why real time is associated with the PCR. So real time means live. That means here scientist gets the opportunity to observe the increase in amount of DNA molecule during the PCR technique. This real time PCR is also known as qPCR, and here the Q stands for quantitative PCR. Okay, and it is actually based on the general PCR technique. That means with the help of qPCR technique we are simply amplifying our target DNA molecule. Okay, or more specifically I should say the target DNA molecule containing gene or target DNA molecule containing DNA sample. Okay, but here the additional step is, along with the amplification, we can also quantitate our target DNA molecule. That is the utility, and that is the principle of qPCR. That qPCR is based on the normal PCR technique or the basic PCR technique, by which we can amplify and simultaneously quantify the amount of target DNA molecule. Okay, so that is the definition of quantitative real-time PCR. Okay, questions may come that why quantitative PCR is also known as real-time PCR. Then you have to write that quantitative real-time PCR allows scientists to actually view the increase in the amount of DNA as it is amplified. Okay, now the question is, how real-time PCR allows scientists to actually view the increase in amount of DNA as it is amplified? Is there any kind of machine or anything else? So here the answer is, as usual, we are here using fluorophore, or I should say fluorescent reporters. Okay, that's I have also mentioned here. So with the detection and quantification of this fluorescence reporter, we can quantify the amount of our target DNA molecule. Okay, this fluorescence reporter is the main player of the are the main player of real-time PCR. That means absolutely here we are using two kind of fluorescence reporter. One is a fluorescent dye, another one is a sequence specific probe. Okay, SSP stands for sequence specific probe. The fluorescent dye that is commonly used is cyber green. This cyber green binds to the minor group of the double stranded DNA molecule. Okay, and in turn fluorescence, and this fluorescence light will gives us a signal, and that can be measured with the help of standard fluorescent measurement instrument. Okay, so let's discuss about the cyber green first. So suppose this is a DNA molecule. So this is a picture of DNA molecule that I have taken from Wikipedia. So here the cyber green, suppose this DNA is our gene of interest containing DNA sample that we are wanting to apply in qPCR. Okay, so here we have also added cyber green in the solution. This cyber green will bind with this portion. Okay, so this portion is commonly known as minor groove and this thing is known as major groove. So our cyber green dye will bind to the minor groove of the double stranded DNA and gives fluorescence light. So what is your interpretation when you comes to know that cyber green binds to the minor groove of the double stranded DNA molecule? That simply means that cyber green only give you a intense fluorescence signal whenever they binds to a double stranded DNA molecule. If they somehow bind with a single stranded DNA molecule or remains unbound in the solution, the fluorescence signal will be very low. Okay, so whenever you are getting a very significant fluorescence signal from the cyber green, then you have to then you can make sure that your cyber green is bind with a double stranded DNA molecule. Okay, so can we say that this fluorescence based real time PCR can only tells us about the total amount of double stranded DNA molecule that is generated by PCR, isn't it? But now the question is, can this fluorescence dye based real time PCR tells you about whether or not your gene of interest that is present in the DNA molecule is amplifying or not? Is there any kind of confirmation? No. So how can you quantitate your pure amplified gene of interest, whether it is amplified or not? So that is the reason another kind of fluorescent reagent we are using, that is our a sequence specific probe. But we are going to discuss about the sequence specific probe within a minute. But here there is another question, that cyber green has some disadvantages too. What are the disadvantages of cyber green? The disadvantage is cyber green not only prefers binding with the minor groups of the double stranded DNA, but it also prefers binding with the GC-rich sequences. Okay, so suppose if a double stranded DNA molecule is generated by the qPCR technique do not contains any kind of minor group that has increased GC-rich sequences, then there can be a possibility that the minor group cannot bind. Okay, so there can be a possibility that the cyber green cannot bind in the minor group. Okay, so as a result false detection, there will be a possibility, okay, of false detection. So for this is the drawback of fluorescent dye based real time PCR. If you are using cyber green, you have to remember this, that GC rich sequence is preferable by the cyber green. Okay, so if your newly synthesized DNA strand do not contain any minor group with GC rich sequences, there can be a chance that our cyber green will not bind with that kind of DNA molecule, and as a result the overall result will be not exact. So for that reason, and for confirming that whether our gene of interest is amplified or not, we are developing a sequence specific probe based real time PCR, where we are using Tachman probe. Okay, and we have to also keep in our mind that this Tachman probe will not bind with the 3' end of the DNA sample, right? Instead, it will bind with the center of the target DNA molecule. That is the main important and most critical part of uh, quantitative real-time PCR mechanism, because most of us will think that Tachman probe will bind with the 3' end, but it is not, because remember, as I have told you, that during the PCR technique we are not using any kind of restriction endonuclease step, right? So your gene of interest along with another unwanted DNA molecules are also amplifying during PCR. Okay, so if you generate the primer such a way that it will bind with the 3' end, so it will bind here. So by binding the probe here, or can you amplify, can you be sure that your gene of interest is being amplified or not? Is it possible? No. So that is the reason, instead of binding our probe at the 3' end, we are binding our probe in the center of the gene of interest. Okay, and as our plan here is not to use that probe to polymerization as we are using in most of the RDT technique. Here the role of probe is quite different. Okay, because so make sure that this is a sequence specific probe, but with the help of this sequence specific probe we are not facilitating any kind of polymerization. Okay, so we don't need to make this probe specific for the 3' end of the DNA molecule. Okay, so that is the thing that you have to keep in your mind. So now let's start the Tachman based probe. So before going to talk about the Tachman probe, I am wanting to discuss with you about the reporter dye, or you can say it is the short wavelength probe along with the quencher. What is reporter dye and quencher? So the probe is something like that. Here at one end our reporter dye of short wavelength is attached, and on the other hand quencher is attached. Remember one thing, there is a phenomenon called quenching is going on between the reporter dye and the quencher, and as a result the reporter dye cannot fluoresce whenever it is closely located with the quencher. Okay, so that means reporter dye cannot emit or sends any kind of signal if it is attached or closely located to the quencher, and the middle portion is the DNA sequence that is complementary to the target DNA sequence. Okay, so let's come here. So first of all we have performed the denaturation step, that is at 93 to 95 degrees centigrade step. Then during the probe annealing, the probe will find out our gene of interest from the DNA sample or DNA template. Now, so this is the Tachman DNA polymerase that we are using. Now when this Tachman DNA polymerase, this is Tach DNA polymerase. Now when this Tach DNA polymerase, by using the complementary strand in fills up the second complementary strand, during that procedure, due to the 5' to 3' exonuclease activity, it will simply cut the quencher and fluorescent probe containing complex, okay, into single nucleotide, as you can see. So again I am repeating this step, that quencher is annealed successfully with our gene of interest. Now Tach polymerase is involved, okay, in extension of the second complementary strand with the help of TNTPs that we have provided, right? So during this extension, this Taq polymerase, with the help of the 5' to 3' exonuclease activity that it possesses, simply cuts the complex between complex of our short wavelength fluorescent probe and our quencher, and as a result they will cuts into, or they will cleaved into short single nucleotides, and as a result the quencher and fluorescence dye is subsequently separated. Now this fluorescent probe can independently fluoresce and sends a signal. Okay, now this signal will be measured by standard fluorescence measurement instrument, and this fluorescence signal will provide us the information about the generation of newly synthesized strand. Okay, and not only it tells us about the generation of newly synthesized strand, but it also ensures that yes, our gene of interest is also being amplified, because remember, by simply cleaving the probe, that is our complex of fluorescence probe or Taqman probe, it with the help of the 5' to 3' exonuclease activity of the Taq polymerase also are able to complete its extension. Okay, so we can ensure that yes, our gene of interest is also being amplified, right? As a result of this amplification, we are getting fluorescence, right? Because in presence of the quencher complex it is not possible, right? So that is how the sequence specific probe based method of real time PCR is going on, and this sequence specific probe based method of real time PCR will tells us about the generation of newly synthesized strand, right? Because if it fluoresces, that means our newly synthesized strand containing of gene of interest is successfully synthesized. Okay, that is the idea. So let me revise the whole thing theoretically now. So real-time PCR will also known as quantitative PCR. Quantitative real-time PCR is based on the general principle of the PCR, which is used to amplify and simultaneously quantify the target DNA molecule. This is called real-time PCR because it allows the scientists to actually view the increase in the amount of the DNA as it is amplified. Real-time PCR rely upon the detection and quantification of fluorescence reporter whose signal increases in direct proportion of the amount of PCR product in a reaction. These fluorescence reporter include dye that include the double-stranded DNA binding fluorescent dye, that is our cyber green, or sequence specific probe, that is our Taqman probe. So sequence specific probe, as I have discussed with you, binding to the major group, minor group of the double-stranded DNA only. In the solution, the unbound dye exhibit very little fluorescence, as I have told you. This fluorescence will enhanced when the dye is bound to the double-stranded DNA molecule. So cyber green has, however, a limitation that it includes preferential binding to GC-rich sequences. Okay, that means newly synthesized double-stranded DNA molecule lacking GC-rich sequence in the minor group will not be detected with the help of cyber green. Now cyber green monitors the total amount of the double-stranded DNA molecule but cannot distinguish between different sequences. That is the reason we cannot ensure that whether our gene of interest is amplified or not with the help of cyber green, and that is the reason why we have to develop Taqman probe. So this is the definition of quenching. Quenching is a phenomenon by which a molecule called quencher interact with the fluorochrome and reduces the quantum yield of the lifetime. So quenching is a phenomenon by which a molecule quencher by which a molecule interact with the fluorophore molecule or fluorochrome and reduces the quantum yield of the lifetime. That simply means that here the quencher molecule will inhibit the fluorescent emission of fluorochrome. In simpler words, so Tachman DNA probe has three elements. One is our short wavelength fluorescence probe. The second one is the one is the short wavelength fluorescent probe. Another one is the sequence that is complementary to the target DNA molecule, and another one is the quencher. According to the definition of quenching, the interaction between the quencher and the fluorescent or fluorochrome reduces the quantum yield or the lifetime. That means in presence of quencher, the fluorochrome cannot fluoresce, and that is the phenomenon that is used by Tachman probe. So this Tachman probe will bind with the designed to anneal at the center of the target DNA. When Tach polymerase elongates the second complementary strand during PCR, its 5' to 3' exonuclease activity, that is the important, cuts the probe into single nucleotide. This removes the close proximity between the fluorophore and quencher, so quencher cannot impart its quenching ability to fluorophore. As a result, the short wavelength fluorophore can now fluoresce and a signal will be detected. This is proportional to the number of newly synthesized strands. So that is all about the qPCR mechanism, how it is going on. Remember, it is known as quantitative PCR or quantitative real-time PCR because it allow us to amplify the target DNA molecule. Not only amplification, but it also allow us to simultaneously quantitate our target DNA molecule. That is why the name quantitative PCR, and it is also known as real-time PCR because it allows the scientist to actually view the increase in amount of the DNA molecule as it is amplified. So the overall thing that is going on in case of real-time PCR is just by detection and quantification of fluorescent reporter. One is fluorescent dye that is binding with the minor group of the double-stranded DNA molecule, and another one is sequence specific probe. The fluorescent dye is the cybergreen. So the drawback that cybergreen exhibits is its preferential binding in the G-series sequences of the minor group, and in case of sequence specific probe, this sequence specific probe is usually Tachman probe, which consists of three elements. One is our short wavelength fluorescent probe. One is our short wavelength fluorophore or fluorescent probe. Another one is the sequence that is complementary to the target DNA sequence, and another one is the quencher. Quencher, when the fluorescent probe is in close proximity, exhibits its quenching activity and inhibits its fluorescence property. But when the Tach DNA polymerase, with the help of its 5' to 3' exonuclease activity, cuts the Tachman probe into single nucleotide, fluorescent probe and quencher cleaved into single nucleotides. So there is no longer closeness between the fluorescence and quencher, and as a result, this fluorescent molecule can now fluoresce and it will send the signal, and this signal will tell us about the generation of newly synthesized strand. That is the overall concept of quantitative real-time PCR.